אני יוסף סידר, זה פרופסור חיים סידר. יש לנו את אותו שם משפחה כי אני הבן הבכור של חיים, ואם אתם צופים בנו, אז אתם מקבלים הצצה לתוך שיחה שמתנהלת בינינו במהלך כל החיים שלי ו-49 השנים האחרונות של אבא שלי. או לפחות מאז ששנינו זוכרים את היכולת שלי לדבר. אוקיי. So, I know some information about your switch from mathematics and physics to biology, but if you can bring me back to where you were as an 18-year-old young man deciding what he wants to do.
So I grew up I was born in Brooklyn. I grew up the early part of my life in Brooklyn. And then my father's work was moved to a small town in New Jersey. And so he picked up the family and we moved out there. And then instead of being exposed to the educational system in New York, I became exposed to an educational system in this small town in New Jersey, which in my opinion, was not as good as what we could have gotten in New York.
And you went to public schools?
I went to public schools.
In Morristown, New Jersey?
Yeah, and my parents were very, very aggressive in the concept of education. They were both basically immigrants. My mother immigrated when she was just a young girl. I grew up in Canada in the United States, and my father was born to immigrant parents. All of his siblings were born before they came to America. And both of them basically had no serious education. My father never finished the 6th grade, and my mother also dropped out of high school later on. And I think because of that, they were very, very concerned about education. They felt that our future had to do with getting a good education. And they really encouraged, aside from just sending us off to school, they really encouraged us. They tried to do homework with us and help us, and they took us to places so that we'd be able to see the world a little bit more and learn from things. I remember going to the planetarium in New York many times to learn about the stars and astronomy and the cosmology and so I grew up in that environment.
But you had a sense or an awareness that you were a good student.
Yes, I always was a good student. My teachers told me I was a good student, and I got good report cards. Report cards like
like my son.
Your son, maybe. Yeah, I enjoyed it. I enjoyed the learning. I had a tremendous feeling of curiosity, which occupied my mind not only during formal studying, but also in normal everyday life. And I couldn't, just going from one place to another, my mind kept thinking of, is there a shorter way, is there a different way, is there a road over here? And I would explore those things. And in New Jersey, when we moved to New Jersey, I had a bicycle. And that was one of the things I did. I would take out my bicycle and I would find these shortcuts and pathways, and it was very exciting for me. So, yeah, and the other thing is that I think in the times that I was growing up, the atmosphere in America was very encouraging to science. I think that the American people understood that the future of the country...
This is the early 50s.
This is the early 50s, right. Understood, we were just coming out of the Manhattan Project, the atomic bomb, and other areas of science.
But how were you exposed to these things as a young teenager?
It was in the news. You know, Albert Einstein, for instance, who I met, by the way. Albert Einstein.
Where did you meet him?
My parents took me. There was an open day at Princeton University. And my parents, who had just gotten a car, actually, drove out there. And we went there, we wandered around the campus at Princeton, and there he was. So you have to understand, everybody knew Albert Einstein. He was in the newspapers, you know. You can't say it was on television, because there wasn't much television then, but it was talked about on the radio. And people, you know, parlor meetings, people would talk about Albert Einstein. Nobody understood what he did, but, you know, he was the genius of the world. And then the whole concept of the atomic bomb was a big issue in the United States. And, you know, it was all made possible by these amazing scientists, these amazing physicists, who not only did their personal work on physics, but also worked together to do this project. And it had an influence, and you know, I was not the only person influenced by it. And so I decided that I'd like to do something in science. I was good in math, and because of that I was good in physics. And so I applied to MIT. And
which was your first choice?
I applied to a number of schools. Almost all of them I didn't get into. I just got into MIT. I applied to Harvard, I applied to Princeton. Princeton I wasn't serious about because I went to visit there and I didn't like the environment at Princeton. It was very, very foreign to me, it was very Christian. And they were serious about it. You know, in those days, every student had to go to church on Sunday. All Princeton students had to go to church. And so I was not that enthusiastic about going to Princeton. And I really didn't know that much about all these different places. And then I got accepted at MIT and I started studying there. And I was good, you know, it was good. I loved it. I learned a tremendous amount. For the first time in my life, I was exposed to other people who were of the same liking as me, who loved science and loved to explore. And it was,
so
it was very exciting.
Your high school didn't prepare you for
it
MIT? It
prepared me very, very well. I had a number of really excellent teachers who they weren't scientists themselves, but they knew how to excite a person about science. In particular, I had an amazing math teacher who really, he took me under his wing and he helped me and he taught me all sorts of advanced things. And I had a chemistry teacher who, he was a little bit crazy, but he really taught the fine points of chemistry and about science. He taught you how to be a scientist. So I had really good teachers in high school, not all of them. I went to MIT. MIT was a fantastic atmosphere for studying.
How so?
Science. First of all, it was very open. It wasn't structured like most colleges and universities. The students were free to come to classes or not come to classes. If you wanted to, you could just take the final exam. And there were students who did that. There were students who came to MIT. And within a year and a half or 2 years, they just took all the exams and they finished. And the teachers were very, very open. You could always go over to any professor, any teacher, and talk to him and ask him questions. And so it was an excellent, really an excellent environment. And it was exciting. We had physics, for instance. So the whole class, everybody at MIT had to learn all the sciences. 2.5 years, everybody had to study advanced math, advanced chemistry, advanced physics. And today, by the way, they include biology. And physics was like going to a Broadway show. You know, all 900 or 1,000 students would come to this gigantic room, this gigantic auditorium. And the people who gave the lectures did it together with demonstrations, which included bombs going off and lights and smoke and, right? And
so for me to understand, is this studying physics in the early 60s or the late 50s in MIT is like studying computer science today?
Absolutely.
It's the new frontier?
Absolutely, yeah. Physics and engineering, all sorts of different kinds of engineering.
And you saw yourself spending your life in that field?
I saw myself spending my life, but as time went on at MIT, I discovered that math, even though I was very good at it, there were people who were much, much better, much more creative, had a much deeper understanding of math than I did. And it discouraged me. I discovered that in physics, I really didn't understand completely the physical concepts, and I was only doing well because I was good at the math part. Mm-hmm. And that bothered me, right? And at some point, I went the way of everybody, and I said, you know, I have to do something. I'm not going to succeed in these things, but and
so you were seeing peers who had a better understanding than you in this area.
Yeah. And it was very important in those days. And it said that this is the way it has been in Israel most of the time I've been here. It was very important to use your education to go out and then, you know, establish a profession, right? Mm-hmm. And I felt that as well, and I wasn't sure I could do these things in a profession. So I decided that maybe I should go to medical school. And
was there any pressure from your parents?
No,
at this point?
My parents were completely open. Not only were they completely open, but there was an economic issue, because I remember the first year at MIT, the tuition was $900, and my parents
for a year.
For a year.
Okay, a lot has changed.
Yeah, and my parents had set up a savings account when I was a little boy to save money for, it was something that the government sponsored. Every month you put in money so that you'd have enough money when your child reached the age of 17 or 18, he could go off to college. And they paid the tuition from that. And then the second year I was at MIT, they raised the tuition to $1,100. And this savings account was not enough to pay for it. So I knew this was an issue. And still, I decided I'm going to go to medical school. My parents, you know, didn't say anything about it. Again, I only got accepted to one medical school, and I got accepted.
You applied to more than one?
I applied to a bunch of medical schools. I only got accepted to one.
Why?
I don't know. And then only.
That's encouraging for many people who are being rejected by good universities.
I suppose so. And so, and I was only accepted a week before the classes started. Evidently, I was on the waiting list, and I was only accepted a week before.
To NYU, NYU Medical School.
NYU, and so I went to.
Wait,
NYU.
but before we leave Boston, I mean, so how many years did you spend in MIT?
Four years at MIT. Again, they were wonderful years.
And other things happened during those years. I mean,
other things happened. I guess I matured some more, and I made a lot of good friends. I got to know people in the community in Boston, which was an asset.
And that's when you first became aware of Rabbi Soloveitchik, right?
Right.
Who was an important figure for you.
Right. Rabbi Soloveitchik, who was basically the leader of what we would call today modern Orthodox Jewry, was located in Boston. And every Saturday night he would give a lesson to the general public. And when I got there, when I first came, and he used to give it in Yiddish. And one of the students from Harvard went over to him and said, you know, if you want to attract some young people, you should do it in English. And so within a half a year, he went over to English. It attracted a lot of people, and I was addicted to this. And
so you'd
I'd
go
never
to
been
hear him
exposed
speak
to
every Saturday night?
Almost every Saturday night, I would go and listen to him speak, and it was a real, aside from what I learned, it was a real intellectual challenge. He was, even though he was a great rabbi, he was a scientist in the sense that you see, in the sense of thinking and his approach and his methodology and what was important to him, right? And how he went about looking at things, perspective. It was very, very scientific, and I had never been exposed to that before, and I loved it.
Mm-hmm.
Yeah.
Okay, so you decide to be a doctor.
So I decided to be a doctor. I went to medical school. I got
a practicing doctor.
Yeah,
that was the idea.
The idea was to go out into the world and have a profession, and so that I wouldn't be a drain on my parents. And the second day I was at NYU, I noticed this big poster that the U.S. government is opening up a new program to support MD PhDs, right? That was interesting, but the most interesting part was at the bottom of the poster where it said that it covers all tuition for 6 years and a stipend, right? So I immediately applied for this. I had no idea what it was. I had no idea if I wanted to do it, but this was a way to get ahead and help my parents, and I did it, and it turned out to be a fantastic.
But you must have known what you were going into. I mean, it couldn't have been only about the scholarship.
I knew what I was getting into in the sense of time, that I would be spending a lot more time doing this, and I was excited about the fact that I would also be doing research in addition to studying medicine. I'd already realized what studying medicine entails. And when I bought all these books that basically the teachers made us understand that you have to know the whole book. Yeah, I was excited about it. And
so starting out at NYU, you already knew that you weren't going to be a practicing doctor.
it was developing. I didn't know.
Mm-hmm.
It developed over time. Yeah. And
were there, I mean, how did the biologists in the generation before you get their training? If this, I mean, the MD PhD was a new program, it was a new concept.
right.
So where did the biologists in the previous generation come from?
So there were people who had studied, I mean, before they opened this program, which is basically a formal program, there were people who had gone to medical school, and because of their medical education, they got excited about something, and they wanted to do more research, and they went on and they did a PhD. Right? And there were people the opposite, people who had done a PhD and realized that if they want to do research in medicine, they need some sort of medical training, and so then they went to medical school. There were less of those. So there were people like that. In fact, the person who was my mentor, Jimmy Schwartz, had done that. Right? But it wasn't formalized, and it was hard for people to do it. I mean, you had to get accepted to these, and you had to find funds to do it. And this made it easy. The U.S. government decided that it's important for doctors to go into research. And that's the future of medicine, you know. We can't, the concept is you can't keep going on giving people aspirin for everything. That you have to develop medicine, and the people that would best be equipped to do that would be doctors. And so that was the concept. Mm-hmm. And they started the program the year that I first started medical school. They started it in 6 different places in the country. I ended up being the first person to finish the program, which I'm very proud of.
What happened to all the rest?
They all finished. They all went through.
Oh, you were just the first?
I was just the first, okay. Yeah.
You, over the years, I've heard you say that science is asking questions. And I'm curious to understand what were the questions that were being asked when you just started out? I mean, a lot of when I hear you speak about your work now, then a lot of this information is taken for granted. Right. So what questions were being asked in the early 70s at this point? Or
so in the 60s,
still the 60s?
Middle 60s, I started doing a PhD. And the laboratory that I went to was, at that time, the big subject in biological research was in biochemistry. And it's very understandable. I mean, there was a concept that in order to understand materials or the world around us, you have to know chemistry. And so if you want to understand biology, you have to know the chemistry of it, right? And there was a great emphasis on learning biochemistry. And I went into a lab like that was studying biochemistry and using microorganisms to study biochemistry. And by the nature of it, we were asking questions that were not, you know, gigantic, earth-shattering biological questions. We were asking more things that had to do with biochemistry, and they were more specific. And I started off that way, and it was very good actually, because I really learned the tools of research, and I learned a lot about how you have to really get down to basics to do research. That you can't really, you must get down to the basic level in order, in the end, to discover something or to do something.
So can you explain that to me with some example from these years?
Yeah, so we were working on this enzyme. That's what I did my doctorate on. And the enzyme, we didn't know completely what it was used for in the cell, right? But it was a new type of enzyme. And as part of my doctorate, I worked out how this enzyme does it. In other words, the actual mechanism for how the enzyme does it. It involved mathematics, which I was good at, right? And it involved designing experiments that sort of challenged the biochemistry, challenged the system, so that you could break up the mechanism into parts. And it was really basic work. We were working on something that I don't think had big philosophical implications, right? But it was exciting to do that, and it turned out that this mechanism that we discovered turned out to be very important for other enzymes and other systems.
What is the mechanism?
So it's complicated. I really can't explain it here, right?
Okay.
But it involves real chemistry, right? How the molecules interact with the enzyme and how they attach two different molecules get attached together, and how the enzyme does that, and what the enzyme needs to do that. And it turns out it's important for many biological processes. So it was a contribution. And then through serendipity and luck and I don't know what, I ended up being in the public health service in the United States.
Wait, so this is your doctorate?
Right.
You did your doctorate in Jimmy Schwartz's lab.
Right.
Was there anyone else who was in that lab then that continued to be a colleague of yours? And
Not really, but I stayed there for another year to do an internship, and I had an internship in pathology, which meant that I could spend a lot of time doing research. And during that year, I worked for somebody named Eric Kandel. You might know the name. Eric got the Nobel Prize many, many years later for his work on neural circuits in a simple organism called Aplisia. And it was the first time that you could work out how a behavior is mediated by the nerves that do it. Very exciting. He was a real pioneer in this. And during this year, I worked with him. And we did some biochemistry on this system that he was developing at that time. And it turned out that the work that I did there was very, very important. It turned out that one of the things that we discovered together turned out to be the basis for what we call learning in this organism, the biochemical basis for this. And I did that with him. He's a very, very exciting and dynamic person, and he really influenced me tremendously. And we're friends till today.
As a scientist, as a teacher?
As everything. Yeah, he was the one that taught me that he was, before he started doing research, he was a psychiatrist. He was a board-certified psychiatrist at Harvard, and I once asked him. I said, you know, I'm young. I'm going out into the world. What do you think I need to be a scientist? I think I phrased the question differently. I asked him what does he attribute to his success, right? And he said, his answer was very promising, very surprising. He said, well, I think the most important thing is to be a psychiatrist. He said, whatever you do, you're not going to do alone. You have to do it with people. You have to know how to talk to people. You have to know how to interact with them. You have to know how to get the best out of people, right? And he said that being a psychiatrist prepares you perfectly for being a scientist. So,
I'm not sure I understand that all the way. I mean, psychiatrists as opposed to psychologists see everything as a chemical reaction.
No, I think his meaning was as a whole, psychiatry, psychology. In other words, understanding human nature was an important part of doing research. And obviously it's not the most important part, but it is a very, very important part of doing research. When I set up my own lab, and I didn't set it up the way he did, but one of the concepts that I had when I set up the lab, a developing concept as he set up the lab, was to make the lab like a family. And so that people share, people talk to each other, and even though all through the years, every student, whether doctoral student or postdoctoral student, had their own project, right? There was always interactions, everybody asking for help from other people, whether it's just, you know, technical help, you know, I'm not going to be here tomorrow, please do this, or they would sit down and discuss things, and I spent a tremendous amount of time with students. That was basically my profession, sitting and talking to students. Yeah, all the time.
And in these, I mean, so now you've worked in one lab for your doctorate, and then you moved on to Eric Kandel's lab. At this point, did you know that this is where you want to spend your life? That this is what you're good at. This is where you feel comfortable?
So yes, basically, I made a decision that I'm not going to pursue the medical part, right? And this actually was a critical thing because when I finished the internship, because of the Vietnam War, every medical student had gotten an exemption to study medicine. And independent of all the other things, the lottery and the draft, anybody who finished medicine had to serve in the armed services, in the Army or Navy or Air Force or whatever. And they took doctors, right? And they sent them to Vietnam, and 70% of my class was overseas, sent overseas as doctors. And I had decided I'm not going into medicine, and that was part of the decision basically helped me get accepted to the Public Health Service. Public Health Service in the United States is a service that provide medical services to all sorts of communities, like the Indian reservations, right? Poor communities, communities that are very far away, and the Public Health Service takes care of that. But they also have a research institute, and I ended up in this research institute. And then for the first time, I was exposed to molecular biology.
Oh, so up to this point, it's chemistry.
It's basically chemistry, right?
So your exposure to what you ended up doing your whole life happened because you were forced to join this organization.
Sort of. Yeah.
Okay, so continue from that point. What?
So this was a lab that was working on DNA. The mentor, Gary Felsenfeld, had studied with Linus Pauling, another Nobel Prize, not just a Nobel Prize. He won the Nobel Prize twice, once in chemistry and once for a Peace Prize. And he was a well-known chemist, and he had gotten his education from him. And I was very fortunate to begin my career in molecular biology that way, because he provided a real basis for understanding DNA, right? I mean, most people understand DNA in the popular way, you know, it's a language, and it can be used to make proteins and build the cell, and, right? But behind this all, there's a basic structure. And he really helped me understand that. First of all, understand how it works, and secondly, understand the importance of it. And I learned a tremendous amount from him on DNA and the structure of DNA. And it was a tremendous influence.
And was this happening in other places around the world?
So
or were you lucky enough to be in the right place at the right time where something absolutely new was being discovered?
Yeah. So there were other people working on aspects of molecular biology, but very few people were working on the structure of DNA, and especially the structure of the proteins that wrap the DNA. Well, that was completely new. There were really very few people studying that. It was completely new. It was in its infancy. In retrospect, we made mistakes. We looked sometimes in the wrong direction. That's part of research.
But if we're going to now talk about DNA methylation, is there something that happened in Gary's lab that put you on a certain direction? Absolutely, yeah. So can you explain that?
So one of the discoveries that we made in Gary's lab was, in my opinion, a key element of molecular biology, a critical element of molecular biology. And, you know, and that is that people were always, people knew a lot about the molecular biology of simple organisms. But in simple organisms, the control of how genes are used is very simple, okay? So, for instance, in a bacteria, all the genes in its genome, on its chromosome, are expressed, are used all the time, except for a few. And for those few, there's a mechanism of a protein called a repressor that recognizes the gene and turns it off. Very simple regulation, right? And
when was that discovered? This simple thing that ends up being your life's work. When was that? That
was sort of the late 50s, early 60s, in parallel with the discovery of the structure of DNA by Watson and Crick.
Okay.
And a lot of this work was done by another pair of amazing scientists, Jacob and Monod. And they did this work, they discovered this, right? But animal cells were different. Animal cells had a much, much, much bigger genome, many, many more genes, right? And it was obviously much more complicated. I mean, you know, a man is much, much more complicated than a simple bacteria. And nothing was really known about regulation. And these were the beginnings. Why? Because it turns out that in animal cells, the major aspect of regulation is that the DNA, the basic source of the language, is wrapped in a structure. And what determines whether a gene is going to be used, is going to be expressed, right, or read, it's going to be read by the cell, is the structure. If the structure is open, then the machinery can go in and read it. If the structure is closed, it can't. And we, using a very simple method, we discovered that this is the case. That indeed, what's important about the control of genes in an animal cell is what's called accessibility. The ability of the machinery of the cell to actually see it. And we made this discovery in Gary's lab, myself, and Gary, and another very, very, very good colleague who was also there like me for medical school, Richard Axel.
Who was doing his army service there
the
the same way you were doing
his army service, right. Mm-hmm. Yeah.
So how much was, just so that I understand this juncture, how much was that, how much of a breakthrough was that in understanding your next step?
For me, it was a very, very big breakthrough.
What did you understand then about the accessibility? What did you think you can learn from this mechanism?
So I understood that now all the other things that we had thought were important, okay, like these repressors, or like the machinery that reads the genes. Okay? In bacteria, the machinery that reads the genes is very important for regulation. It's not the case here. The RNA polymerase, which reads the genes, right? Really, it's just a machine. This is the way I thought of it, right? What's important is getting into the right place. So all those other things became secondary for me. And what was important now, which I understood, was the structure of the DNA. Right? And yeah, that was a very, very big influence. And when I left that lab and we came on Aliyah, we came to Israel, and that's what I devoted the lab to understanding
So it seems like, and just the way you're describing this, if to a certain point it was you trying to get through school and find a way to pay back your
right.
university tuition and making your parents feel comfortable with their need to give you a good education. And at this point you got excited about an idea and everything else moved aside?
Yeah, basically. Exactly.
Because you knew that this was, this was something that will affect all areas of medicine, of our understanding of how.
I don't think I looked at it that way. First of all, I was much more tsanua about it. But I saw this as a challenge for me that I thought was very important. But I really didn't think about the overall big implications of it.
So how many years were you in the...
2 years.
Just 2 years?
Yeah. I finished my service and then we came, me and you and Mommy and
those 2 years were in Maryland?
Yeah.
In Washington?
Right.
So you finished your service and you knew that you were going to move to Israel?
Yeah.
I've always wondered, what else was... what other option did you have then? I mean, I always assumed that the pretext to your marriage to Mom was that you're moving to Israel as soon as you can. Were there other options? I mean, were you giving something up? It sounds like you're on this fantastic path with the best people in the field.
Right. So looking back, I think that Aliyah was a very, very big element. And I mean, I saw almost all of my options as moving to Israel. I saw that as a very, very important point. And I must admit that the professional aspect was secondary to that. It was high up, but it was secondary. Mm-hmm. We were very excited about the idea of coming on Aliyah. And Mommy, for her reasons, because she had basically been completely absorbed in the Zionist idea since she was a baby. And me, because I was just learned about it late in life, and I was very excited about it. And it's interesting that you ask about that, because the issue of the professional aspect of it didn't occupy my mind, but it occupied my interaction with other people. When I finished in the public health service, so you know, people who left there went out to look for jobs, or to go either to continue in medicine somewhere, which a lot of people did, or to find a laboratory or a university to go to. And I had decided I'm coming to Israel, and I had arranged with somebody at the Hebrew University that there would be a job for me when I came. Mm-hmm. I came here. It was much easier then than it is now.
Who were you speaking to here?
And there was somebody in the Department of Biochemistry that sort of took me under his wing, and Shimon Gat, and he helped do all this.
I always imagined you coming to a blank slate, that there was nothing here when you came. But was there an infrastructure for the science that you were going to?
A wonderful, wonderful infrastructure that was not rich. It was not rich like in the United States. It was much different. Many more things were shared. You didn't go out and buy everything you needed for the lab. Right? But as I've said before, I probably told you before, I was warned by my colleagues that coming to Israel was a sacrifice, because there's less money here, less facilities. But the most important thing that they warned me against is the idea that Israel's a small country. It may be good research there, but it's a small country. And so when you go and you come in your field, which is also already something that not many people do, you're going to go to Israel, there's going to be nobody there, nobody for you to talk to, nobody to interact with or to collaborate with. And a mistake, a big mistake. You're going to Israel, you know. And because we were so suffused with the idea of Aliyah, this didn't affect me. But years later, I thought retrospectively about this. I came to the conclusion that they were wrong. And they were wrong for a very simple reason, which, I don't know, I think is an obvious thing, but maybe not everybody's aware of it. But they were wrong because of the Israeli student. The Israeli student is really something special. And I slowly discovered this, that there's something about the Israeli students who come to the lab, who at the stage, at least at the stage of their career where they want to do advanced work in a field, in biology, and they just had all the properties that you want from a student. You know, they had motivation, and they had background, and they had this thinking ability, and they knew how to ask questions. They were nudniks, you know. They really, you know, pushed the hell out of me. They really, and it was a real asset that, you know, I didn't know about before, and I was very, very happy to discover. And until today, I mean.
And you can see a real difference between how students interact with their mentors in an Israeli lab or in labs elsewhere in, you know, European or American labs.
Yeah, absolutely. There's a different a different approach.
So there's an Israeli mentality that comes out in a lab that doesn't exist.
Yeah, I think so. I like to call it culture. Science is culture. You know, you can't. I think you can't really, taking somebody and putting him in a university and teaching him biology 1 and chemistry 1 and physics 1 and mathematics and that doesn't make a scientist. There's a culture. And to be a scientist, basically you have to get it from from your culture. A culture that includes, curiosity, asking a lot of questions, being critical, giving credit to other people. Right? You can't do science without knowing what other people did and understanding that this is good and this is good. Maybe this is bad, but understanding that you're not going to start from invent the wheel again, right? But there are people behind you. It's an understanding
that doesn't sound very Israeli, if you ask me.
Yeah, it's true.
Everything else did, but that one, where I think, at least in my area, you get a sense that the good, innovative Israelis think that they are inventing the wheel.
Yeah, so that's why I've
over and over again.
So that's why I specifically said students,
okay,
who had not yet reached that point. You're right.
I understand. So tell me about like the first steps in Israel, because I've always been, if I put myself in your shoes, and I imagine the kind of momentum that you were in, it seems very scary to start in a place where everyone is warning you, you have no, like there's no way to really advance.
I came here, and I admit it was hard, and for all the reasons that people warned me about. It was hard to get money.
But you had a position, you had a lab.
I had a position. Mm-hmm. I was very fortunate. I applied to the NIH for money in America, and even though there were all sorts of things going on in the government then that didn't allow funding, but in the end the funding came through. And I became rich and had money to do.
So you came here with a grant?
Basically, I mean, I got the grant half a year or a year afterwards.
So where did this myth that I grew up with come up, where you almost became a bicycle shop owner?
Oh, yeah. So, getting grants, the naive scientist feels that, if he writes a grant and he gets it, that's it. He's set for life. Right? And it doesn't work that way. You have to justify yourself every 3 years, right? And rewrite and reinvent and rediscover, and and it's a very big, it's a lot of pressure to do that. And so at one point, for a long period of time, when I wasn't sure whether I'd have enough money to do the kinds of things that I want to do, I did say that. I said, look, I don't get grants. I'm going to leave the university and I'll do something else. And since I loved bicycles, that was...
Okay, so, but you came here with a grant.
Because when I first came, the first day that we unloaded my bicycle from the lift, right, everybody at the port came over to look at this new bicycle.
It was a 10-speed bike.
It was like a 10-speed, relatively fancy bike, right? And so I said to myself, oh, if I don't get my grants, this is what I'm going to do. Mm-hmm. I'm going to expose Israel to these bicycles. Yeah, sorry, I interrupted.
No, that's... sometimes I ask myself if what would have happened if you would have been the first person to import a 10-speed bike. So you came here. You had a grant. The university gave you a lab. Can you tell me about that? Maybe, I mean, I'm curious to understand how this is the first time where you're running the show, right? This is already your lab. It's a reflection of your ideals, your values. So I'm curious to understand what that was like, but also maybe if you can lead me into the scientific questions you were asking at this point.
So everything went very slowly. I think this is general true of research. And I started working on a project, and it turned out the project was successful, but not very meaningful, right? And we started some other projects, and some of them turned out to be good.
But all this was in the area of DNA regulation.
All of it was in the area of DNA regulation.
This
right.
was your thing.
Right.
You already knew that.
That's what I wanted to do, and that's what I was good at.
Did the term epigenetics exist then?
It existed because it had been used previously in works on genetics and in development, but not at the molecular level, only at the conceptual level. Meaning, okay, so you do an experiment and you discover that people would discover that some trait, whatever it was, would it be an animal or a fly or a worm, whatever they were working on, and they would discover that they can't explain it by the genetics, right? And so they would say it has to be related to genetics, right? And they called it epigenetics. So in that sense, the concept existed, right? But at the molecular level, it there was no way of understanding it. Right.
So you knew that this phenomenon exists, but you didn't understand how it works.
I didn't, right.
Okay.
Right.
So now, if you can lead me into what resulted
right.
with DNA methylation.
Okay, so just to put everything into perspective, I think the major question was as follows, okay? That's the way it started off. Somebody, DNA had been discovered in the structure of DNA in the late 50s. And in the early 60s, they discovered the basic dogma of molecular biology, that DNA, you make RNA from it, and from that you make protein, right? All that was discovered. And then, at some point, it turned out that somebody discovered or mentioned, I don't even know if it was a discovery, that if you take a look at a human body or an animal body, if you take any cell of the body, it has the exact same DNA. And that makes sense because the body comes about from a single cell, and the cell divides and copies the DNA, and divides and copies and divides and copies. So every cell of the body has, and so somebody really showed that in every cell of the body is everything. So the albumin gene, which is used in the liver, that gene is in every cell of the body, right? And this posed a big question. If the DNA, if the information is in every cell of the body, why is it that albumin is only made in the liver? What's the control? How does it work? Right? And that's what started the whole thing. Okay? And the realization that the same DNA everywhere is what triggered it.
And this realization was only happening now. It's not, because the way you describe it now, it sounds like the first question you'd ask.
Right, but only the concept that every cell has the same DNA, right, was relatively new.
Okay.
Or relatively thought of new.
I understand.
And so we basically started asking that question. And there was somebody at the Hebrew University, Aaron Razin, who had been working on methylation, the modification of the DNA by methylation, but he had been working on it in a bacterial virus, a phage. Right? And it plays, in the phage, it plays some sort of auxiliary role, not really very important. Not even important for the phage itself. A phage can do without it as well. But this was an interesting thing, that you could modify the DNA, right? And I don't think I understood the full implications of this, but it was something that I realized this is some sort of addition to DNA, something that could add information to DNA. And so we started working on it. And one of the first things that we saw was that it took a while for the methodology to develop. Some of it we did, some of it other people did.
Can you talk about the methodology in a way that I understand?
So you have to be able to measure methylation, right? And to measure methylation, you need tools. And slowly tools became available by chance, right? An enzyme, for instance, that will cut the DNA, but if it's methylated, it won't cut it. Mm-hmm. So this gives you a way of knowing whether it's methylated at this place, right? And then further techniques of that nature, right? And so one of the first things that came out of this new curiosity was, indeed, if you look at the albumin gene, right, and it's methylated everywhere in the body, in all the different tissues, DNA, it's there and it's methylated, but in the liver,
except in the liver,
it's unmethylated. Right? And if you take any tissue-specific gene, you find the same thing. You know, hemoglobin, right? Crystalline from the eye, exactly the same thing. So this confirmed the idea that, and it looked like that methylation was serving as some sort of repressor, and that this was the way it was controlled.
What year was this when you got that confirmation?
This is 1975, 76, something like that. 77, around then. Yeah.
And then was there one paper that you wrote that somehow captures the essence of this?
So the question then arose, and I must say that there are critics until today, right? The first question that arose is does methylation do the repression? In other words, okay, it's present on genes that are repressed, right? Fine. It's not present on genes that are expressed. And but is it the methylation that does that? Mm-hmm. Right? And
this was the question that you were asking.
This was the question that I was asking. Okay. Yeah. And we created a system for testing this. Again, we had to develop the technology to do it, but basically it was based on the idea that you could take DNA and put it into a cell in culture that's growing on a plate, right? And that DNA would get integrated into the rest of the DNA of the cell. Mm-hmm. Right? And then it would express. So what we did was we took DNA from a gene, we put it into the cell, and we saw that it expresses. And then we took this same DNA, and using technology that we developed for this, methylated it.
And it wasn't expressed.
And put it into the cell, and it wasn't expressed. And so this was a very, very big turning point. And at the same time, in the same experiment, we discovered that once something is methylated, it will remain methylated even though these cells divide and divide and divide and divide. So that meant that this mark is a memory device.
But this sounds like like a later understanding.
It took, all these things took time. As time went on, we understood more and more what, right?
So the first question was, all cells have the same DNA. How is it that they're not expressed all the time? The answer was apparently they're methylated, or the methylation is what determines whether they're expressed or not. The next stage was realizing that it leaves a mark that stays or can be affected.
They went together. One went together with the other. Yeah. Yeah.
And at this point, were you understanding the implications of this?
We were beginning to understand the implications of it, yeah. And I must say that what held us back from being bolder about this, I'm talking about our personal view of it, not bolder, but what kept us back a little bit was the constant criticism that we got from other scientists. That,
was there one specific paper that was criticized? What?
No. The problem is that what I described before about the discoveries of Jacob and Monod, about how genes are regulated by proteins, by repressors, right, was really considered a basic element of biology. And it was hard for people to overcome the idea that maybe there's something more than that. Okay? And until today, there are critics. I run into it all the time when I write a paper and I send it in. I get criticism about that. But now I think it's mostly well accepted that yeah, these proteins play a big role, there's no question about it. But if you want to remember something, if you want to remember a decision, and this remembering turns out to be a very, very important thing in a complex organism like man. Okay? Because if you can make a decision at one time during development, right, and then lock it in place with methylation, you don't have to worry about it anymore. Right? And in an organism where it's so complex, you know, genes on and off and partly on and partly, this is a very, very big advantage. And so this memory idea, and this is basically epigenetics. This is the basis of the concept of epigenetics.
Okay, so you sometimes use an analogy to an annotated text or an annotated book. Maybe just for the few viewers who are watching us who don't know what DNA methylation is, maybe you can use that analogy to explain what it is.
Yeah, so, you know, this explains part of methylation, maybe not the whole thing, but so I like to make an analogy to other texts. I like to look at DNA as a text. It is a language for all intents and purposes. It has, as opposed to Hebrew, which has 22 letters, this language has 4 letters, but it has words, and it has sentences, right? And you can say that it has paragraphs, and even I would like compare the chromosomes of our cells to like a volume of an encyclopedia, right? So we have 23 different volumes in our cells, right? So it's a text, and
that exists in its entirety in every multiplication of the cell.
Exactly right. Now, other texts, you know, like a language text, like Hebrew or English or any other language, right? Many of the languages have methods for annotating the text, okay? So for instance, in French, you can put an accent mark or not put an accent mark, right? But the best is Hebrew, right? So you have the text, the letters, and sometimes it's very hard to read without the vowels. In the Torah, it's written without any punctuation, right?
Or separation between words.
Or sometimes separation between words, right? So the text is very hard to read. And in Hebrew as well, in terms of the Bible, and there are instructions of how to read it, where to put the accent, where to put the emphasis, right? Where there should be a stop between words in a sentence, what's called Tamei Mikra. Right? And those are all annotations that help the reader read the text. Because otherwise, and you can change it, right? And we just had, for instance, we just had in Parashat Yitro, we read the Ten Commandments. And when you read the Ten Commandments in the Torah, there's different annotations, right? As opposed to when you would read from the Bible, from the Torah, just read it personally, and maybe it doesn't change the meaning so much, but it changes the overall perspective of what the Ten Commandments are, how they should be grouped together, right? The concept, the real concepts that they're trying to express, right? And that's annotated. And methylation does exactly that. By methylating the DNA at different places, you basically annotate it so that the cells know how to read it. And it's an important part of language, an important layer of language, and especially in higher organisms like man, plants, for instance. And it's a necessary element, especially in long-living organisms, where you need this kind of memory, right? And methylation is very, very important for that.
Can I just ask something about how we remember these marks? They're never like when are they? When do they start? When do they cancel out and start over again?
Yeah. Well, great question. Wonderful. So this is one of the definitions of epigenetics. Okay, so we obtain our DNA, the text, we obtain from our parents. All of our genes we get directly from our parents. Okay,
without the annotation.
And it comes from, originally it comes from the sperm and from the egg, with the annotations that were in the sperm and the egg. But very quickly after the first conjugation of the cells, and you start getting a few divisions, all of the methylations erased, right? And then as the part of development, the organism, using information in the DNA, builds a new methylation pattern. And this was an important point because one of the big things that we have to be able to explain is, right, how do our cells, how does our DNA know how to make this pattern, right? There's a whole side area of methylation that's amazing and very, very important.
So this seems to me like a whole other layer of the original work that you did.
Right.
And so when did you start understanding this?
So this occurred in the 90s. We started understanding this was important. We published a couple of key papers in this explaining how simple things get established. But until
how
today,
it's erased, and then what determines the new methylation?
Yeah.
Is that, am I using the right words?
Yeah.
Too?
Yeah.
I heard you give an example. I think this is the continuation of this layer of your work. You said that a pregnant woman during her pregnancy undoes her genetic self to become someone new because of something that happens to this annotation while she's pregnant.
I don't remember saying that, but
is it not true? Is it, or what I think you said is that a pregnant woman goes through some kind of reshuffling of her genetic or of the methylation that results in a different person, a mother.
Yeah, so that's true. This is really later work that we're doing, which is fascinating. And
so it's not a direct continuation of understanding how it's erased and reset.
No, but
okay,
it's still a part of it. This is something that's really we're doing now, which is really new, and which I think is the future of what we call epigenetics. Okay, and that is that it turns out, so the way I presented it to you, it looks like all the changes in methylation that take place during the development of the organism. And then the person is born, right? And that's it. Now he has fixed. That's the idea of methylation. It's all fixed. A liver will remain a liver forever, and a kidney cell will remain a kidney cell forever because of methylation, right? So it turns out that things in our environment, or things that we do, or can also influence our methylation pattern in different tissues of the body. So what happens is what, like you say, right? Something in the environment will change this basic methylation pattern so that the liver is now directed in a little different direction. Right? Becomes adapted to something that happened. Okay? So we know that the diet, this is all done in mice, but in a diet, when the mouse goes over from a maternal diet that it gets from the blood to a milk diet, okay, there are tremendous changes in almost all the tissues of the body on how those tissues now behave. Then when the young pup becomes weaned, again, additional changes. Right? And one of the things is what you mentioned, we now know, didn't know this before, that when a woman becomes pregnant, okay, this brings about, it's probably from the hormones that are used, this brings about a change in the liver, in the pancreas, in the brain, in fat cells, in muscle cells, brings about a change in their methylation pattern. In other words, not just a transient change in how those tissues behave, but something that will then be permanent.
And how far is this from your initial assumptions in 76 or 77?
We just never thought about it. There were dreamers that said that this is true. There are still, today you see a lot of it. People say, you can't explain schizophrenia, for instance, by genetics. Must be methylation, right? Mm-hmm. Or you can't explain, or diet. If you eat this kind of diet all the time, it'll change your methylation pattern and you'll be a different person, right? For good or for bad, whatever, right? And so there's a lot of hypothesizing about this, right? But now we're beginning to get the first data that shows that it's probably true. To what extent, we don't know, right? And there's still a lot of work to do on it.
Okay, so that, I think it's impossible for me and impossible in this amount of time to really get into like a detailed understanding of how one thing led to another. But maybe if we can leave the pure science for a second. One of the questions that I ask myself about your work is the analogy to an annotated text is difficult for me to translate into something that's chemical, because an annotated text has meaning. And the person who wrote the text had an intention, and the annotation either changes that intention or gives it its meaning. Is there something that gives meaning to methylation? Or another way of asking that, are we just very complicated machines that work automatically, or is there some purpose for these annotations?
Yeah, I think there is a purpose for it. This is what we call in science teleology, right? And teleology meaning that basically it's an idea, but it's something that can't be proved, right? Because you're asking about the system, you're asking a why question about the system. And so this is not really science, this is more teleology, right? But yeah, I think we can guess. And my guess is that if we look, we've learned a little bit about the evolution of methylation. I mean, there are many organisms, simple organisms, that don't have methylation, right? And there's some organisms that have a little bit of methylation. And over the time, we've learned how this methylation got established, right? And it seems to me that it basically is a way for adaptation. And adaptation that's needed in long-living organisms. It seems to be. And there might be, right, this is a little bit, it sounds far-fetched, but there's already data to indicate that this thing, it might be a way, mechanism for evolution itself, okay? And the idea is that there are now experiments that, it actually started with experiments in man, which show that the experience, the environment of the parents, okay, can influence the behavior of the offspring. Mm-hmm. Okay? So the classic example is this number of studies that have been done on the famine that existed in Holland during World War II. And if you look at the offspring of these people, people who were subject to starvation for very long periods of time, right? These people have eating disorders. And the research is actually very good, I mean, to show that it's not a matter of growing up in a house where somebody, you know, preserves food all the time, but it's a real effect. Even if you separate these people from the offspring, from the original parents, it still shows up. Okay? And
but I know from hearing you, voice opinions about similar things that you're critical of, like the direct connection between a phenomenon that we see that has some social significance and a very simple way of putting it into a context of evolution.
Right. You put it very mildly. I'm very, very critical. But I think that's just part of my scientific nature that, doesn't get convinced easily. I have to be proven. It has to be proven.
But despite the criticism, what you just said suggests that there is something, like if there is any kind, if I understand what you said, that this is a why question, but if there is any meaning behind methylation, it may explain our ability to adapt to different things.
Yeah, no, I think the bottom line is that and I'll just give you an example, okay? Again, I don't know anything about the molecular aspect of this, but there's an experiment to show that if you take a mouse and you expose him to a odor that he's never seen before, and you condition him to be afraid of the odor, mm-hmm. okay? And then you take that mouse and he fathers pups. Those pups will be afraid of that odor. Okay? There's a good chance that if this is really true, that this is mediated by methylation. Very good chance. Okay? But the significance of it is that this, if it's right, is a shortcut to evolution. Okay? Because how does evolution work? Evolution waits for mutations, changes in the DNA, and then the selection of the environment, right, amplifies that organism. Right? But if there's a mechanism for the parents to learn something and transfer it epigenetically to the children, and then maybe another couple of generations, we don't know anything about that, right? This is a real shortcut to evolution.
So that's the meaning.
That's the meaning, right?
And that's a tremendous concept. It connects the chemistry of it with something that is right. much more abstract and right. cultural.
Yeah.
Can you talk a bit about the applied science that is a result of your fundamental research?
Yeah, so there's been a lot of stuff. there are now drugs that are used for cancer that take advantage of the fact that in cancer, every form of cancer, there's abnormal methylation, and these drugs are used to treat on the basis of that, to change the methylation, and it doesn't work for every tumor, but it works for a lot of them.
So we've had a whole conversation about your work without saying cancer once.
Right.
But the main application of your work has to do with understanding cancer.
That's, yeah, it could be. That's, and then the most recent thing is, I think, is going to be a revolution in medicine, I think. And this is work that was done in our department by somebody named Yuval Dor, who, and basically this is a subconscious transfer of ideas and working together. And what he's done is he takes blood from anybody, and it turns out that in the blood there's DNA. Now, of course, there are cells in the blood, and the cells have DNA. But in addition to that, there's free DNA. Where does this free DNA come from? It comes from cells in the body that are dying. And why are the cells dying? There's something wrong with them, okay? And they die. And they release the DNA into the blood. So, of course, every DNA is the same. It doesn't matter whether it came from the brain or it came from the muscle or it came from anywhere, right? And it's the same DNA. But what Yuval did was said, oh, but the DNA that comes from different tissues has different methylation. So by using methylation, we can determine where these pieces of DNA are coming from, and therefore we can detect cell tissue illness, tissue abnormality. Right? And this can be used as a diagnostic tool. In many cases, it can be used as early diagnosis for almost any disease. Almost all diseases involve death of cells. It can be used for cancer. It can be used for monitoring whether treatment is helping in cancer. It can be used in multiple sclerosis. It can be used for predicting heart attacks. It can be used for predicting this thing that the football players suffer from, this traumatic encephalitis. You pick it up on DNA much earlier than you see the signs. Right? So extremely, it's a real revolution. If it works out, it's going to be a real revolution, and all based on DNA methylation. And yeah, it's an amazing thing. Yeah.
So I've heard from the people in the university who are in charge of taking research and finding a patent out of it, that they're all very upset with you for not doing that, not working with that in mind. Do you have something against applied science?
I have nothing against it. Every year I get a call from the university about if I have something that I want to, but my mind just doesn't work like that. You know, this whole concept of the diagnosis, it was not my idea. Came from somebody else who's perhaps much more minded with that. It's just not the way my mind works, and I love doing basic research. And it never works in the way, well, how can I use it?
But the way you fund the lab is by explaining how it affects our life. So the further away from these, from the pharmacology of it, the harder it is to explain what you're doing is essential.
Very, very true. It's hard to obtain money for basic research. No question about it. Yeah. Absolutely.
Okay, so maybe just like before we end this conversation, which seems like it just began, because so many things remain not perfectly clear to me in the work itself. but just so that I have a chance to ask some questions that otherwise I wouldn't have a chance to ask. Over the years, are there some principles that you found to be not only effective, but a reflection of who you are that come across in your work or in the lab with students, with just the way you conduct yourself during the day or towards a goal in a project?
Yeah. So I already mentioned before that I like to treat my laboratory as a family. And we really are a family. We celebrate things together, and we do things together, and we talk all the time together. But there's another thing that I do that I think it's just a part of me, but I think it's an important thing, and that is that I insist. Not only do I sit with students a lot of time, I think that's an important part of science. I mean, I rely on them, but when you sit with a student, he learns more, I learn more from him, and it enriches the science. But I have a principle that no data in the lab, there's no data in the lab that I haven't looked at. And every piece of data that's generated in the lab, I sit down and I go over it. And sometimes it takes hours to do this. But I never let a piece of data get into a paper or get into a talk or even have the students talk about it without first going over it in detail and analyzing it from all aspects. And the reason is that being a good scientist is tricky, not easy. It's easy to be fooled. It's easy to make mistakes. It's easy to go in the wrong direction. And we're living in a time when there's a lot of scientific fraud, and a lot of it comes from this, not insisting on going over everything very, very carefully.
I think one of the things that are a mystery to me is that it seems like you have a work discipline that is almost inhuman. Not because, I mean, everyone has periods in life where they work hard. But my impression from the way you worked is that it was never hard. But you're somehow able to work continuously and concentrate. Where does that come from?
Yeah. So I must admit to you that I try not to bring home problems. Right? So there were many times when I didn't get a grant, or a paper wasn't accepted, or a student did something stupid that I just don't. I come home as if it never happened. Right? And I did that partly because I value the home as family, as peaceful family, right? That should be separated from this. Right? And partly for myself that, okay, this is a problem, but I don't necessarily have to make other people suffer from this problem.
So is there a support group that helps you with,
with
this pressure?
We should set up a support group for
if the family is not where you allow these pressures to come out, so where do they come out?
I'm not sure. It probably comes out in the end in the family, and just not specifically. Yeah.
I think I was asking about just my image of you at home. At work you work, but my image of you at home is always with some article in front of you. Where do you get the concentration to devote so much energy into your work?
It's a disease.
But it's not from the content or the substance?
It is, it's a substance, but the disease of constantly wanting to learn more and read more and study more, and it's a compulsion or an obsession.
And is that somehow part of why you've been able to make these discoveries?
I'm not sure. I don't know. But I certainly, I'm always amazed by the fact that usually new ideas come up when you're reading some article. And the article could be far removed from what you do or what you study, right? And it just clicks an idea. And so, you know, it becomes addicting. If that works, it's great.
Okay, so maybe just the last thing, since I know I won't have another chance to ask this question in front of other people. So I'm 49, you're 75. If you look at what, you know, what each stage in your life was able to produce, is it more difficult now? Is it different now? Is the experience valuable? Are you slowed down?
Yeah.
I mean, what happens to a scientist at this stage in his life?
So it's very individual. And I must admit that I'm very conscious of this question all the time. And I keep saying to myself, right, that I don't want to do what other people do, other people who I don't think is the right way, and I constantly work on myself. So it's a conscious effort.
An effort to do what?
To make this stage in my life appropriate. Okay, so I've slowed down, and I spend more time now in the house, and I spend more time eating meals and being with mommy, and I come in later to the lab. Of course, I leave later, but that doesn't make any difference. And the research in the lab is much slower. I'm much less of a tyrant than I used to be with students and other people. Right? The direction of the research has changed into...
But is the understanding still as sharp as it always was?
No, I don't think so. But there's a perspective that you get from a lot of experience that's more valuable than the sharpness. So it's an important aspect, and I see it all the time. I spend a lot of my time working with young scientists who just came to the university. And I won't say helping them, but talking to them and being with them and sharing with them. I enjoy that very much, and I think having this kind of perspective helps. So yes, I'm conscious of the fact that things are changing. I'm trying to build my life so that, or continue my life in a way that the kind of skills that I still have, where I can make meaningful. At the same time recognizing that, you know, my analytical abilities are nothing like what they were. Sometimes I sit with a student and she starts talking and I have no idea what she's talking about because it's just too fast. Right?
I can say from my perspective, I've noticed that in the last couple of years you spend more time explaining your work to people who otherwise wouldn't understand. So from my point of view, this is a good stage. I'm finally getting to understand what you've been doing all these years.
And it's interesting that you say that, because doing that has helped me in my research. You know, explaining things more, and especially to people who understand it less, right, has really had an influence on my perspective. So, yeah, it works both ways.
Okay. Anything else that you think we should talk about?
Well, that was amazing.
Okay.
Really amazing.
תודה.